Thank you for an excellent talk. We're now going to have a quick question and answer session. So could I ask the speakers to please go to the table? And we're going to have 15 minutes because we're running a little late. And so there are 2 mics at either side of the room. And so if you have a question, could you please go to the mic and say who you are and where you're from, etc. Thank you. Okay, so are there any questions? Okay, if we go over there.
Is it on? Okay, yeah. This is a more general question, and I'm not trying to pick on one area of research versus another, because I use everything from cells to rodents to primates to humans. But from the epigenetic perspective, it seems that the processes would be quite distinct between a short-lived species versus a long-lived species. Rodents have to be immediately adaptable to their environments versus humans have to be a little bit more malleable. They have to adapt over generations. But I may be right or wrong on this, but I'd like to know what the comments from those experts on that perspective of species differences. And Rob might want to weigh in as well. So.
Yeah, who had more than one species in their talk? That would be me. I wasn't expecting that one, Kevin. Yeah, so I think this plasticity thing, I think, you know, one thing that has really occurred to me when I talk to people who study rodents, us included, and even brain experts included, is that the developmental pathways that we're looking at, extrauterine versus intrauterine, are so much different. So when rodent studies are being done for breastfeeding, for example, and that may be a pattern of development that actually happens in utero. And so whether these things can be sort of merged at the end of the day, because so much of the organ-specific changes are dictated by which species you're looking at, I'm not sure. That's not a good answer. Sorry, not so good.
Okay, I have a question for Caroline. You showed us your model of how maternal BMI allele score, you know, by taking that into account, that you could conclude that you had strong evidence that maternal BMI is causally causing changes in child DNA methylation. But I'm just wondering, how does Mendelian randomization rule out the alternative pathway that the maternal genetics, you know, are partially inherited by the child, and that it's the genetic differences that are then causing differences in methylation?
We condition on maternal genotype.
But what do you mean by that exactly? You said the BMI score. So I mean, how do you condition across the entire genome?
No, we condition on the alleles used for the in the Mendelian randomization framework. Right. We don't condition for the entire proportion of genetic variation across the entire genome.
So there's a large chunk of maternal genetic variation that is being inherited by the child, which are not accounting for.
So I didn't have an opportunity to include in my talk some GWAS of methylation data, which we just will be submitting for publication shortly. And what we've done now is, obviously we're really interested to know whether what proportion of variance in methylation is explained by underlying genotype. And one of the analyses we've undertaken in that is using GCTA, or ask the question, of all the SNPs that we know are associated with BMI and with a host of other traits, what proportion of those SNPs are associated with methylation, or the MQTLs? And interestingly, we see that very few methylation SNPs are linked to or explain variance in BMI and adiposity, where we see a much greater proportion of methylation-relevant SNPs are implicated in height, in type 1 diabetes, and rheumatoid arthritis. So there's an interesting difference across different traits as to the proportion of genetic influence on epigenetic patterns. So
that's also going to be conditioned on the tissue that you're looking at, because this is all in blood.
Absolutely,
too, right?
Yeah.
Okay.
I'm interested in the difference between male and female offspring, that is, as the fetus develops, and I think even in the case of sperm, is there a difference between Y-bearing and X-bearing sperm? And I would think, Carolyn, in your situation, you could actually separate male and female. I would think the sperm could be, I would think the monkeys could be. So the question is there a difference in male and female offspring?
In terms of genome-wide methylation differerences between sexes, we see a huge number. We haven't looked at it in relation to adiposity per se, but we've identified using just a straightforward epigenome-wide association study approach, and we see something like 17,000 statistically significant hits. So we see, and these are in autosomes, not in sex chromosomes. So, and we think some of them are associated with sex hormones and
so there are differences between
there are huge differences between girls and boys, essentially, in terms of the distribution of the methylation patterns. How that tracks through or is associated with subsequent traits or diseases, we can't say at the moment.
X and Y sperm?
So we haven't really segregated my X or Y containing sperm. We've done it as just one big mix. The only thing I can tell you is that from the offspring that we've examined in terms of an IVF setting, we have found we have not found any statistical difference in terms of any of the methylation profiles that we've been addressing.
I'm John Carroll from downstate in Brooklyn. Maybe I shouldn't be asking questions because it's speakers. I should yield my time. We have so little time.
No, please.
It's okay?
Yeah, no, we're fine.
Caroline, I was really impressed by your very careful presentation of your data and the concept of bashing BMI, which I'm all in favor of doing. I wonder, with this wonderful ALSPAC study that you have, and obviously you had banked tissue, it seems, whether you've considered correlates of the BMI, in other words, much more relevant ones, that is metabolic disease phenotype rather than just this idiotic sized phenotype that everybody's fixated on.
So we have data on
the kids. I mean,
yeah,
the offspring,
on the kids.
right?
We have DEXA data and fat distribution data at multiple time points, which we've begun to look at. But the beauty of BMI, particularly in the context of something like Mendelian randomization, is that you can then leverage huge amounts of publicly available genotype data. So once you know your SNP or variant that's associated with methylation, you can go and look that up in Giant, where there's 100,000 individuals, and you get leverage a huge amount of power in doing that. So yes, we are looking at fat distribution and other more subtle measures of adiposity. We've also got, about ready for release, metabolite profiling on lipids on about 200 metabolites. So looking, and another postdoc in my group has done some reciprocal causal analysis methods to see whether lipids are altering methylation or methylation is altering lipid profiles. So there's a huge swathe of work that's possible there, but as Rob's rightly pointed out, it is very much limited by the fact that it's measures in peripheral blood. But I think certainly in the application of Mendelian randomization, one of the reassuring features emerging is that the genetic correlates of methylation seem to be quite highly consistent across tissues. So if you were to identify a genetic correlate of methylation in blood, it's quite likely that it is reflective of other tissue types. So, but we're still developing the methods.
We have such tissue correlates, but we're looking forward then to a dysmetabolic phenotype from you. Thank you.
Quick question for Jacob. You showed in obese women that breast milk leptin concentrations are dramatically elevated, and of course you're aware of neurotrophic effect that leptin plays in the early postnatal mouse in hypothalamic development, that work of Richard Simmerly. I'm just wondering, are there any data that have tested whether leptin in humans serves a similar role affecting brain development?
I don't think anybody's dared to measure baby leptin surges the way they have in humans, but I can tell you that those, in the context that I'm using it as an exposure to the infant gut, that if you knock out the leptin receptor in the infant gut, there is a dramatic change in the phenotype in terms of IBD. So there possibly is a role, because the gut is so porous to these hormones, transversing and avoiding the acid environment of the stomach in the early infant, and then reaching the receptors at the level of the lamina propria, for example, and then playing a role in educating the immune cells and bacteria that live down in there.
So for those of you who want to have lunch here in-house, there's a cafeteria on the 3rd floor. For speakers, sponsors, and planning committee members, Allison will provide you with a lunch ticket, and we'll reconvene at 1.
And I think we should just thank all the